Good day. This is our protein synthesis video about the explanation of the problems of the portfolio. Our team members are Valeria, Jimena, Pérez, Contreras, Gael, Patricio, Gaytan, Botello, Yvette, Rodríguez, Vargas, Sergio, López, Balderas, Ricardo, Andrés, Véliz, Salgado, and myself, Pedro, Cortés, Almazán. We hope you enjoy.
Well, this is the first part, the first question that is about sequencing, and we had to briefly describe the process we use to decode the messages. Well, to decode these messages, we in first instance we have to transcribe like the DNA to RNA, pass it. Doing this is by placing the corresponding like the anticodons in the given ones like that are corresponding to the DNA ones, and then identify or translate these codons to the corresponding amino acid. For example, like when CUU, if you have a CUU codon, it corresponds to leucine amino acid, and do so with all the codons presented. Finally, with the help of the abbreviations, like for example, in the leucine that I previously mentioned, the abbreviation is LEU. Then we search on match in the symbol table, and we found that the corresponding symbol for each like amino acid. So then we translate it or trespass it to a message that it's a sentence in English at the final.
Okay, so now we have the question, what is the role that the codons play in protein synthesis, and what is used in coded messages? Well, first of all, we have to explain codons, which are the sequence of 3 nucleotides in DNA or mRNA that specifies a particular amino acid during protein synthesis. They're normally found in the genetic code, which is the basis for establishing how genes are encoded into DNA proteins. So this is basically, the codon is basically the part of the process in which proteins are made. And it is specialized in that scenario. Normally, the language that is using coded message is conformed by letters, which are also known as mRNA, which is a single-stranded DNA strand that is complementary to one of the DNA strands of a certain gene. As I mentioned earlier, well, each process needs a code. And to represent each code, we have letters, and that's what we use to represent the codons, the coded messages. And yeah, that's it. Okay, so now we have what 6 letters would not appear in coded messages. Give a reason for your answer. This question was not so easy, but it is because due to the fact that each type of abbreviation of a certain type of code comes because it is abbreviation from specific amino acids. So the letters that you will not find are P, J, O, U, X, and Z. This is because there are no amino acids that has this abbreviation.
First, in the procedure, we need to write the mRNA chain complementary to the DNA sequence that it is provided down, and upon finding the mRNA codon match each triplet of the DNA base by base. The mRNA is already started and we just completed it. The underlined ADN to the RNM. For this exercise, we just simplify substituting using this cheat table that is from G to C, from A to U, to T from A, and from C to G. Now, from the previous step, using this two charts, we need to find the name of the protein. The chain is already started, and from the previous 3-letter bases, we changed the name. Now, to end this step, we need to just look for the symbols of each of the letters of the proteins. As you can see, the protein chain was not that large, and it made just a little or simple code that was make my day.
Now we came to the part to decode the message. First, I changed from ADN to ARNAM, and it was ACU, GCU, AAA, GAG, UGA, UGU, GCG, CGC, and GAA, which translated into THR, ALA, Liz, glue, stop, cis, ALA, ARC, glue, which translated into the message, take care. Next, I did the same steps, which translated into CAU, GAA, UAA, AUU, UCG, UAG, GCA, UAA, UCA, CCG, UAU, which translated into his, glue, stop, ILE, sir, stop, ALA, stop, sir, pro, fear, which translated into the sentence, he's a spy. The next sentence was AUG, GAG, GAA, ACU, UAA, AUG, GAA, UGA, GCA, ACA, UAA, UUU, AUA, GCU, GUC, GAG, which translated into the sentence, meet me at 5.
Next, we did the same in this case, and here we can see how we translate from DNA. Then below is the RNA codes. Then we pass it through the amino acid, the corresponding amino acid, and we only put the abbreviation here, TIHR, his, glue, and the stop codon said stop, and so on. So then translating, after doing that, like here, that is the transcription, then the translation, and these abbreviations, by searching the symbols, the resulting message was, the plan has changed. Then we do the same here. We can look the first group of codons if corresponds to DNA, then the RNA, then the corresponding amino acid of each for each codon that we solve with the help of the charts and tables that provide us in the portfolio. And then we do like these steps, like in this scheme, that from DNA to RNA and the transcription to the amino acids. And the message was, the ship sails at dawn.
Okay, so how do you know which databases must use when you transcribe the DNA sequence to mRNA codons? You can know the bases adenine, thymine, guanine, cytosine, and uracil. When did you use them? Because adenine is always paired with thymine or uracil, and guanine is always paired with cytosine. So in transcription, a single strand of DNA is transcribed to RNA with a respective base pairing, except RNA does not contain thymine. Instead, it uses uracil.
Suppose predict the DNA sequence for the first message in part B began with TGT, CGA, CGA, TGA instead. Is the message changed? Why or why not? Well, since we replaced the 4 triplets, then the only letters that will be changed are the first 4 letters of the message. Then, the first triplet only changes the last letter, since the T translates to A and G to C. The first letter is not changed. After that, the next triplet is unchanged, since it's the same code. Next, the third triplet is the same as the second triplet, meaning that the second letters repeat. Finally, the last triplet is the same as the original first triplet, meaning that the first letter of the message is the same as the fourth. After finding all those relations, we can conclude that the message changes from take care to tat care.
Okay, so now we have the question. Suppose some codons to different amino acids are mapped. What would be the effect in your translation of coded messages? What will be the effect on protein production? Well, the translation of the coded message will become inaccurate, and copies of several different proteins will be created, so they might not work as well as well-coded protein. They fail because the proteins formed will have different composition due to the fact that there's not a specific or single amino acid given to the codons. So yeah, basically, if the process is not done correctly, or there is a certain type of change, what it will cause, it will change the whole process around, and it will not work as well. I mean, it could produce some proteins, but they will not work as well as a well-done protein.
During the real production of protein in a cell, what could happen to our RNA chain before leaving the nucleus? After the RNA is processed, it carries the instruction to a ribosome in a cytoplasm, so before the RNA leaves the nucleus occurs a translation on the ribosomes, which consists of RNA proteins. Then RNA helps bonds form between the amino acids, producing a phosphate chain. What step could after transcribing the DNA to develop a more complete model of protein synthesis? Translation is a step that is used to make a complete model of protein synthesis after DNA is transcribed. Because DNA is first transcribed and converted into mRNA, which carries codon from the protein synthesis. Thus, mRNA is translated to form proteins. Protein synthesis takes place at ribosomes. The one step is translation.
Finally, we had to create our own message. First, we started with the DNA. TCA, CGC, CAG, CTT, ATT, TGA, GTG, CTC, ACT, GGG, GAA, CGT, TTA, CTC, and TGG. Then we translated to mRNA, which is the things we can see here. And finally, we have all the amino acids that could be translated to letters, which are serine, alanine, valine, glutamic acid, and then we have space or stop. Then threonine, histidine, glutamic acid, we have another space or stop, and the last word would be proline, leucine, alanine, asparagine, glutamic acid, and threonine, which translates to save the planet.
Thank you very much for your attention.